Hello, my name is Keila, and I'm here to introduce our Biology 135 Lab Section 11 presentation by Carson Huber, Emma Holder, and Keila Jellings. So I want to start out with the introduction by saying that all living organisms need proteins, and in order for these proteins to do the jobs that they're specifically given, they need to be activated. And a process that they go through in order to activate is post-translational modification. Now, there are many forms of this, but the ones that our lab focuses on is going to be ubiquitination, which is the process of adding a protein to another protein. And the specific protein that we're focusing on today is ubiquitin, which is a protein that contains 76 amino acids, and it is used as a marker to the substrate that is attaching to. In classical ubiquitination, it goes through three processes in order to get that ubiquitin attached with a covalent bond. The first one is the E1 step, which is the activation of the enzyme. Then it goes to the E2 step, which is the conjugation that catalyzes the transfer of the ubiquitin. And then E3, which is ligation, the final step where the ligase catalyzes the formation of the isopeptide bond. In classical ubiquitination, often used by the eukaryotic cells, are ATP driven as their energy source. But there's actually another type of ubiquitination that is NAD dependent for their energy source, and we call that NAD dependent ubiquitination, which is often carried out by pathogens. And so what pathogens do is, instead of doing this, they go through like the E3 process by using an E6 protein, attaching it to an E6AP protein. And what it does is it really messes up their host cell's process of ubiquitination. It really messes with the cell division and causes things such as cancer. And a good example of this would be the HPV virus. The protein that we're focusing on for this NAD-dependent ubiquitination is Rab33B, and we're going to see how the use of the addition of NAD will carry out the reaction between ubiquitin and the Rab33B, which will create a protein called Rab33B-UB. And with any ubiquitination process, with both of them, it can go through a reversal process called deubiquitination, which utilizes enzymes such as deubiquitinase to break the isopeptide bonds that are created between the ubiquitin and the protein that it is attached to. So for our lab, we'll be using DUPB as the enzyme that is going to break the isopeptide bond, between the Rab33B and the the ubiquitin. So it will turn Rab33B into two different products, which is Rab33B and ubiquitin, which is the original proteins before ubiquitination occurred. And so, that is our quick introduction, and I'm going to hand it over to Emma.
Hello, for this lab, we were examining the forward and reverse reaction of the ubiquitination of the Rab33B protein. We did this in two parts. So for experiment 1, we were looking to see how the presence of NAD plus affects the forward reaction, and we hypothesized that if NAD plus is not present, then the reaction will not occur because NAD plus is the energy source for this reaction. And for experiment 2, we were looking to see how the presence of DUPB affects the reverse reaction. And we hypothesized that if DUPB is not present, then the reaction will not occur because it is the catalyst for that reaction. The purpose of these two experiments is to see how the hydrolysis of high-energy chemical bonds drives biochemical reactions, as well as looking and understanding how post-translational modification of proteins such as ubiquitination occur, and how the components of ubiquitination contribute to the process. So the materials and equipment we used for this are 4 test tubes, a centrifuge, the SDS page gel, an electrophoresis apparatus, and an incubator. And the various solutions we used were the Tris HCl, DTT, NaCl, SDEA, NAD+, a buffer solution, RAB 33B ubiquitin, DUPB, water, the RAB 33B ubiquitin complex, a protein ladder, as well as the gel stain, which is 1% Kumacy Brilliant Blue R250 plus 50% methanol and 10% glacial acetic acid, and the destaining solution, which is 40% methanol and 10% glacial acetic acid. So the procedures for experiment 1 and experiment 2 are pretty similar. The first thing you do are create the reaction mixtures. So for reaction 1, you're going to have 2 test tubes, and in both you're going to add the 50 micromolar of DTT and 50 microliters of Tris HCl. And then to the first test tube, you're going to create the first reaction, which contains 10 microliters each of the SDEA, ubiquitin RAB 33B, sorry about that, and the buffer solution, plus 10 microliters of NAD+. And the difference between the first and second test tubes is that the second one does not contain NAD+. So you are going to tap both of them to mix, and then centrifuge for 10 seconds to thoroughly mix. Then allow the reaction to proceed at room temperature for 30 minutes. After that, you're going to terminate the reaction by adding an additional 10 microliters of buffer and heating at 100 degrees Celsius for 5 minutes. You're then going to load the solutions into the SDS page gel. So you are going to load 20 microliters of each. So protein ladder goes in the first well, and then reaction 1 in the second well, and reaction 2 in the third well. You're then going to insert this into the electrophoresis apparatus and set it to 150 volts for an hour and allow the gels to resolve. After an hour, you're going to remove the gels and submerge them fully in the stain for 20 minutes, then rinse with warm water to remove the stain from the gel other than the protein, and then put it in the destaining solution overnight or until you take the pictures. For experiment 2, it's going to be similar, as I said earlier. You're going to create the reaction mixtures, this time with 10 microliters each of the Tris HCl, 50 micromoles of the DTT, and 10 microliters of the NaCl. And then you're going to add the solutions to create the reactions. For the first test tube for the second experiment, you are going to add 10 microliters of buffer, 10 microliters of the DUPB, and 10 microliters of the RAB 33B and ubiquitin complex. And to the second test tube, you are going to substitute the DUPB for water. So everything else will be the same. And again, you're going to tap to mix and centrifuge, and then allow the reactions to proceed in an incubator, this time at 37 degrees Celsius. Then you're going to go ahead and terminate the reactions the same as last time. Load the solutions with the protein ladder in the first well, the first reaction in the second, and the second reaction in the third well. And then you're going to insert into the electrophoresis apparatus for the same voltage and time, remove, stain, rinse, and destain, all the same as the last procedure.
So for the results slide, the picture on the left is the figure 1 from our first experiment. It is an image of the gel electrophoresis of the reaction NAD plus SDEA plus ubidequin plus RAB33B, and then RAB33B-UB, and AMP and SDEA. The first lane is the protein ladder, and the next lane is the one from the test tubes containing NAD+, and the third lane is from the test tube containing no NAD+. And on the right, we have figure 2 from the second experiment. It is an image of a gel electrophoresis of the reverse reaction of RAB33BUB, DUPB, and then it turns into RAB33B plus DUPB plus PRUB. The first lane is on the protein ladder. The first lane is the protein ladder. The second lane is from the test tube 1 containing no DUPB, and the third lane is from the test tube 2 containing DUPB. So we go down to our discussion. And here we have the results. So for experiment 1 in the gel ladder with the BNAD, RAB33UB is found. This is not found in the gel ladder without BNAD. There's also less UB and RAB in the gel ladder with the BNAD. This shows that the BNAD is necessary for the ubidequination of RAB33B using SDEA. This supports our hypothesis that NAD is the energy source of the reaction and that it cannot occur without it. For experiment 2, in the gel ladder where there is DUPB, there is almost no RAB33B-UB. This is because the DUPB deubiquinates the RAB33B-UB into regular RAB33B and ubidequin. We can see this in the data where there is lines for the ubidequin and the RAB33B, and those lines get darker and thicker, which shows there's more of it there. This supports our hypothesis that the DUPB is the enzyme specific to the RAB33BUB, and this also proves the other hypothesis that the DUPB catalyzes the reaction. Our data supports the information we have learned in the lecture about ubidequination and deubidequination.